quality control or QC will be our topic. It's the second step to measure and analyze using an automated clinical chemistry analyzer. We will cover the following point: a definition for laboratory quality control program, the basic requirements for instrument quality control program, quality control product definition and types, a quality control procedure and results, quality control statistics, and Levey-Jennings charts, and finally the quality control management and artificial intelligence. So the quality control program of an automated clinical chemistry analyzer is considered as a part of the total quality control program of a clinical laboratory in a hospital or a medical center. There are two requirements for the running of a quality control program in a clinical chemistry analyzer. Firstly, we need a regular testing of quality control products along with the patient samples, and then the analysis of the quality control results to a specific statistic limits or range using software. So there are two requirements: the running of the QC products with samples and the analysis of the result using a software. The quality control product could be either internal quality control product manufactured by the manufacturer of the analyzer, or could be external quality control product manufactured by a third party.
Our
Now
healthcare
we'll
system is a
watch
lot like the human body.
a small video which talk about the quality control program, if it's external one or internal one, and here the video will be specified about the external one, and especially about the use of the serum indices which are manufactured by a third party in order to evaluate the performance of our clinical chemistry analyzer.
There are a lot of moving parts, each with a specific purpose, and without any one of them, the whole machine would come to a grinding halt. Some of these parts, however, are a little more vital than others. In humans, it's the brain, the spinal cord, the heart. In medicine, it's you, the lab. Every result you provide to doctors is crucial because it may help to diagnose diseases, recommend procedures, and prescribe medication. That means every time you step into the lab and put on your gloves, you're stepping up to bat for the indisposed, the hurt, and the ailing. As such, it's essential that you strive for the best possible results day in and day out, which means maximizing your lab's efficiency with pre-analytical interference monitoring. Luckily, Biorad has the solution: LiquidCheck Serum Indices. Designed to monitor instrument responses for hemolyzed, icteric, or lipemic specimens, LiquidCheck Serum Indices helps identify a leading source of pre-analytical variation, HIL interference. When your instruments begin to lose their touch, be they old or simply in need of service, finding out too late could affect your lab's efficiency. By giving you advanced warning the moment an instrumentation issue is detected, LiquidCheck Serum Indices lifts that weight right off your chest and allows you to breathe easy again. Of course, to unlock the full potential of LiquidCheck Serum Indices, you'll need to surf your way over to the one-of-a-kind Unity Interlaboratory program. With Unity, you can track instrument performance using several easy-to-read instant reports. Even better, you'll have the capability to compare these results with those of similar instruments worldwide. It's a place to teach and it's a place to learn. And when you combine the power of peer assessment with LiquidCheck Serum Indices, it won't be long before your lab waves goodbye to pre-analytical HIL interference. Make a difference. Improve laboratory analytical performance by including serum indices as part of your individualized quality control plan for detection of pre-analytical interferences, and Unity for peer assessment and data management solutions. Together, they are a powerful combination, and they're waiting for you.
So as we have seen that the requirements will be, as we said, the quality control product, the running of the quality control product, as well as a software in order to analyze the results and decide if the results are reliable or some actions should be taken. Here we can see an example of the analysis of a QC results for the serum indices. Now we'll start with some definition prior to start with the talk about the quality control product. The first definition is the statistical process control, which is a set of rules used to verify the reliability of the patient results. It's based on the statistics calculated from the regular testing of the quality control products. Then we'll have a definition for the quality control. So what is a quality control? A quality control in a clinical laboratory is a statistical process which is used to monitor and evaluate the analytical process that produces a patient result. Now we'll talk about the quality control products, what they are and how they are classified. Quality control products are patient-like material, ideally made from urine, serum, or spinal fluid, and composed of one or more analytes of a known concentration. So we know the concentration of the analytes inside this patient-like material or quality control product, and we run this in order to evaluate the performance of the analyzer and if the patient results are reliable. So it has two forms. It can be either a liquid or lyophilized, which means freeze dried, and we have talked about this in the previous what is a lyophilization process and what is a lyophilized product. Also, quality controls should be tested in the same manner as a samples. So we should run them in a similar way to running a patient sample. Now we'll talk about the classification of the quality control products and the procedure as well and results. There are many methods to classify the quality control of products. This classification could be done based on the manufacturer of the quality control product. If it's made by the manufacturer of the chemistry analyzers, this will be considered an internal quality control. If it's manufactured by a third party, this will be considered as external quality control. Also, another type of classification is the quality control range value. If it's the value it's normal or abnormal, or sometimes we could call it levels, level 1, 2, 3, and 4, and so on. And also depends on the analytes type, which will be tested, for example, chemistry, serum, or coagulation, blood gas, immunoassay, hematology, and so on. Also, another scheme of a classification is the quality control published value, if it's assayed or unassayed. And finally, a classification could be done based on the quality control results, if it's quantitative or semi-quantitative or qualitative. Here is the first type of classification scheme, which is based on the manufacturer of the quality control product, if it's the manufacturer of the analyzer itself or a third party or external manufacturer. The external quality control products are made by specialized medical laboratory reagents manufacturers such as Biorad, ADB Diagnostic. They are used to evaluate objectively the performance of a chemistry analyzer. What mean by objectively? Usually the manufacturer of an analyzer will make a quality control which will be a little bit suitable for its analyzer, and this will be considered semi-objective or partially objective, whereas the external quality controller manufacturer will be considered purely objective in this case. And another type will be the internal quality controls, which are manufactured by the manufacturer of the automated clinical chemistry analyzer itself. The application of the external quality control products, we apply them in order to make, as we said, a comparison with the internal quality control, which are made by the manufacturer of the analyzer itself in order to evaluate the performance objectively of this analyzer. Another application will be in the case of we are planning to purchase a new clinical chemistry analyzer. So we use external quality control product in order to make an objective and accurate evaluation for and the performance for the different suppliers who are shared in this bid. So we use it for to make an objective evaluation of the performance. Now we will watch a small video which talk about the third-party control, and this will be for Biorad.
Welcome to Biorad's Blackboard training. This is a series of short, informal tutorials brought to you by industry experts. Today we'll introduce the concept of third-party controls. The testing process begins with a patient specimen. The specimen is sent to the lab for testing. How do we know that the results from the testing process are valid? While running patient samples, labs also run samples of a known value. These are commonly called controls or QC. Controls allow the laboratory to monitor its testing systems and eliminate errors that would impact patient results. For controls to work correctly, they must act like a patient sample. For example, a blood control must be in a blood-like material. This is the same when testing spinal fluid, urine, or whole blood. Controls are shipped in liquid, freeze-dried forms, frozen, refrigerated, or at ambient temperature. Controls are manufactured as a result of one set of operations, known as production. Each production run has some variability, so it's assigned a unique lot number. Concentrations of analytes can vary from lot to lot. Control manufacturers produce many types of controls. Some controls are assayed, meaning that the values are published for all instruments. Assayed controls only give the lab initial target values. They must determine their own values by performing parallel testing on their new and old lots. Unassayed means that the values are not published. Laboratories must also determine their values for unassayed controls by performing parallel testing of new and old lots. Another way to categorize controls is quantitative or qualitative. A quantitative control is used for tests that are reported numerically. A qualitative control is used for tests that are reported with a statement like positive or negative. In the US and in some other countries, regulations state that if you run a test, you must run at least one normal and one abnormal control that day. Many labs recognize that once a day may not be enough for their lab, and they run controls multiple times a day. The lab's defined acceptable range is determined by the initial testing of normal and abnormal controls. Some easy calculations are performed to come up with a mean and SD for each control. SDs, or standard deviations, sound complicated, but if you review our Blackboard videos on statistics, you'll find that they're really quite simple. Regular testing of QC material creates a QC database that the lab uses to verify the test system. This verification occurs by comparing daily QC results to lab-defined ranges of QC values. There's a number of ways to determine if a control is acceptable. The most common approach is Westgard Rules. If you're interested, check out our Blackboard.
So this is one of the classification scheme, as we said before, which will be based if it's an external quality control product or internal quality control product. Usually the external quality control product are more specialized. Also, as we said before, we should consider the matrix effect when we are talking about a quality control product by comparison to the patient samples. Now we'll talk about the second classification scheme for the quality control products, which is based on the value or based on the range. If it's a normal range or abnormal range, as we can see here, the normal range and abnormal range for the potassium analyte. Here also what known as a QC log, which shows the recording of the quality control values for the same quality control, for example, here in the case level 1 and level 2 for the potassium analyte at different dates. Now we'll talk about the quality control procedure or QC procedure. So in the running of the quality controls, absorbance of quality control will be measured and multiplied by the calibration factors, and we have the results of the quality control. As we said, so this result should be lie within a specified range and should be analyzed using a software, and we'll talk about this in the statistic part. So the range is determined by the laboratory through a process of running the QCs on its own machine and until at least 20 times to arrive at the mean as a standard deviation, till such time the manufacturer prescribed range on the QC insert may be used as a guide. So the range is to be defined on the equipment in order to detect the outliers easily. This usually, as we said before, we have two types of clinical chemistry analyzers. We have the standalone chemistry analyzers and we have the modular clinical chemistry analyzer. In the case of the standalone, is usually as this range is which used to detect the outliers is determined or selected manually or sometimes automatically, whereas in the case of the modular platform of the chemistry analyzer should be either entered entered automatically without the need of the interference from laboratory technicians. So after each calibration, normal and abnormal quality controls should be run and the result documented. As we said, this should be the second step prior to measure and analyze. If the values lie within the range, we conclude the analytical system is good and we proceed to testing the patient samples. So the running of the quality control is to ensure that our analytic system is good and the patient results are reliable before reporting them to physicians and doctors. So this is the procedure of the quality control. So first we add the reagents, then we make mixing and incubation at a 37 degree. After that we add the control similar to the way to add a sample. We make a mixing of the mixture and incubation at 37 degree, and finally we read the absorbance as we said before, and we multiply it by the calibration factor in order to have the QC result. Now we have the result. What we should do here come first of all we should know what type of result and how we should analyze the result, analyze the result in order to determine if our analytic system is good and the results are reliable and we could report them to the healthcare givers. So when a diagnostic test is performed in a clinical laboratory, the outcome of this test is a result. It can be either a patient result or a QC result. As we can see here, these are the quality control products, and here is the sample which is drawn from a patient. The result, as we said before, could be a quantitative, a number, or a qualitative, a positive or negative statement, or semi-quantitative, which is limited to a few different values. The aim of the QC results, as we said, either to validate an analyzer is operating within a predefined limits, which means that the analytic system is good, or inferring that the patient results are reliable and we can report them. Now we'll talk about the analysis of these quality control results. In order to make analysis, first of all, we should have a definition for the mean, the standard deviations, and how to use them in a Levey-Jennings charts and the Westgard rules in order to decide what type of error we have and how we can fix this error. As we will see, we will have two types of errors. We have a systematic error and we have a random error, and each one will be related to a specific reason or some reasons or causes. First of all, the mean is the laboratory the best estimate of the analyte's true value for a specific level of control. As we can see here, this is the formula for the mean. Here is an example of the mean. This example will be for the quality control for potassium for a normal range. Here are the recorded result. The quality control has been run for 7 times. So we add the quality control QC values and we make the average for them. Then the second parameter which is needed to be calculated is a standard deviation, which is the statistic that quantifies how close the numerical values, the QC values, are in relation to each other. Here we can see the standard deviation equation or formula. As we can see, firstly we should calculate the mean, then we can calculate the standard deviation. Here also an example for the same results of the potassium. So the same result of the control, we can firstly calculate the mean, then we calculate the standard deviation. So these are the way how to calculate. This is the formula and how to calculate the standard deviation. As we said, firstly we should calculate the mean, and then we use this mean in order to calculate the standard deviation. So now we have the value of the mean and the standard deviation. So, what will be the meaning of these values? Firstly, we should also define the precision, which is a term often used interchangeably with the standard deviation. And precision is used to express how far apart the numerical values or the QC values from each other. Another definition will be the repeatability. The repeatability of a test may be consistent or inconsistent. Consistent means low standard deviation or low imprecision, whereas the inconsistent means high standard deviation and high imprecision. We should have a consistent repeatability of tests. Because, for example, in order to prescribe any medication or to follow the course of the treatment, we should have a repeatability, a good repeatability or consistent. For example, I will take an example, the renal dialysis treatment for a patient. In order to monitor the, usually such patient will have 2 to 3 sessions per week, renal dialysis sessions per week, in order to have removal of the metabolic waste and urine products from our body. So in order to evaluate the effectiveness and the appropriate of the treatment or appropriateness of the treatment, we should make a testing of the patient blood samples. We should make 3 types of we should make a pre-treatment, intra-treatment and post-treatment measuring of a specific analytes should be requested in order to evaluate the effectiveness of the treatment for this patient. So as we said, the values of the quality controls, we should make from the values of the quality control, we should make the calculation of the mean and the standard deviation. Firstly, we calculate the mean, and after that we can calculate the standard deviation, and this will help us to determine of this of our analytics system is good and reliable. So this will be translated in a statistical way, which is called the precision and accuracy. Now we'll use the standard deviation and mean using a Levey-Jennings chart in order to determine what type of error we have. If it's a systematic error or if it's a random error, and we'll talk about this in the next slides. So standard deviation is commonly used for preparing Levey-Jennings with the mean. So we have firstly the mean, and then we have a plus minus standard deviations values. We have a plus minus 1, plus minus 2, and a plus minus 3. And we record the quality control values at different periods. Sometimes we should perform quality control maybe daily or twice per day. So Levey-Jennings chart is used to graph successive quality control values, as we can see here. Also, it should be created for each test and level of control. As we said, we have different types of analytes, and we have also different levels of control for each analyte. We have, for example, as we said, normal, abnormal, or level 1, level 2, level 3, level 4, and so on. So for each level of test, we should have a Levey-Jennings chart. So what is a Levey chart? Is a graph that quality control data is plotted on to give a visual indication whether a laboratory test is working well. So it's named after S. Levey and E.R. Jennings, who in 1950s suggest the use of individual control chart, that word, in the clinical laboratory. We have to access the x-axis and the y-axis. The x-axis represents date and time, and the y-axis represents the distance of standard deviation from the mean. This is the acceptable range. We should have plus 3 and minus 3 from the mean. Some laboratories use plus 2 and minus 2 SD distance from the mean, whereas the acceptable will be the plus 3 and minus 3 SD distance from the mean. So this is called the decision limits. So the decision limits in some lab are plus minus 2 SD, and this is considered not the best choice. The best choice will be the plus 3 minus 3 SD distance from the mean. So as we said, first we run the control, as here we run for 7 times, and then we should have calculate the mean value, then we calculate the standard deviation value, and we use the mean and the standard deviation to calculate the to draw or to plot these values in a Levey-Jennings chart. So this is a Levey-Jennings chart for the previous control, which is a potassium normal control product. Around 99.7% of all QC values shall fall between plus minus 3, whereas in the case of plus minus 2, only 95.5 will be fall within this range. So as we said, the decision limits should be plus minus 3 standard deviation distance from the mean, which is preferable. Only about 0.3, which means 3 out of 1000 will fall outside the plus minus 3 decision limits, and any value outside of plus minus 3 should be considered or associated with a significant error condition, and patient result in this case shouldn't be reported. When the QC results are plotted, an assessment can be made about the quality of the run. As we said, then the technologist, in case of a standalone clinical chemistry analyzer, performing this test should look for systematic error or random error. Whereas in the case of the modular clinical chemistry analyzer platform, these should be made the decision if this is a systematic error or random error, and The reason behind this should be used using an artificial intelligence, and we will talk about this in the last slides. So systematic error is evidenced by a change in the mean of the control values. So systematic error can be either classified as a trend error or a shift error. This trend, in the case of the trend, we have a gradual change in the, and we call it a trend. And in the case of the shift, we have a sudden or abrupt change. So in the case of the trend systematic error, a gradual loss in the reliability of the testing systems. So trends are usually subtle, as we can see here. There are many causes or reasons behind this gradual change in the standard variation value of the QC value. One possible cause would be the deterioration of the halogen lamp. Another reason be the change of the temperature inside the incubation chambers for the cuvettes or reaction cells. This is especially correct in the case of the enzymes, or only in the case of the enzymes. Another reason could be the deterioration of the light filter integrity, accumulation of debris on a sample reagent. Also, as we say, you can see gradual. As we said, this is a gradual change in the value. Another reason, gradual deterioration of calibration of controls materials or aging of the reagents itself. Now we'll talk about the second type of the systematic error, which is the shifts, which is represented in the case of abrupt changes or sudden changes. Or the reasons could be different. For example, before we said a gradual deterioration of the lamp as the result of aging of this lamp, whereas in the case of shifts, we should have a sudden failure or a change in the automated chemistry analyzer light source, a sudden change in the incubation temperature chamber, major automated chemistry analyzer maintenance. So when we perform a major maintenance for the analyzer, so this could be result in a shift or shift systematic error. Also a change in the reagent information and reagent lot and so on. So in addition to the systematic errors, we have the random errors, which is random error is any deviation from an expected result. This could be a positive or negative deviation. Also, this could be acceptable or unacceptable random error and defined and quantified based on the standard deviation. The random error usually, as well as the systematic errors, will be analyzed using the Westgard rules. These rules are made based on an article published by Dr. James Westgard in 1980. These rules could be used either individually or in combination with each other to quantify the quality of the analytic runs in order to determine if we have a systematic error or a random error. So we can use these rules with the artificial intelligence in the case of a total laboratory automation, and we'll talk about this later. So Westgard devised a shorthand notation, as we can see here, for expressing the quality control rules. Most of the quantity can be expressed as an NL, which N means the number of the control observations, where L means the statistic limits. And here are some the 6 basic rules. We have additional rules, as we will see in the summary table later. So these are the 6 basic rules. This 1 2s, 1 3s, 2 2s, 4 rs, 3 1s, and 4 1s. For the first rule, which 1 2s, a single control observation outside the plus minus 2 limits, plus minus 2 SD limits, only about 4.5% of the quality control result fall between two and the three standard deviation limits, which will be this green area and this white area or zone. So random error or systematic error may present. So these rules are help us to determine or decide if what type of error we have. It's a systematic error or random error. And with the use of artificial intelligence, we can also decide more specifically what type of error we have. So these are the basic 6 rules. In this table, we see the most of all of the Westgard rules and meaning also what will be detected. As we can see here, the first and the second rule, the reason could be a random or systematic error, whereas the third and the fourth rule, as well the fifth, we will have, for example, a systematic error, or random error. Okay, so these rules help us to determine what type of error we have of random or statistic. So this is the reason behind the using of the Levey-Jennings charts with the Westgard rules in order to decide what type of problems we have and how how to find solution. So what are the reasons for random errors? Usually a change a calibration lot or reagent lot, recalibration or analyzer maintenance, major analyzer maintenance, or could be to change in a temperature and the humidity of the surrounding environment of the clinical chemistry analyzer. So this is the main target behind the quality control. As we said, this will help us to decide if our analytic system is reliable or not, and if we can the patient results are reliable and could be reported in order to decide if we prescribe medications, or if we have make diagnosis for certain condition, or even to decide or a plan for to make a plan for a treatment based on the values of the of analytes in our blood. Finally, I will talk about the application of artificial intelligence or AI in a clinical chemistry analyzer. The application of artificial intelligence can be seen in the case of the clinical chemistry analyzer. For example, in case of the autoreflex, which is one of the features of the clinical chemistry analyzer. Also, the use becomes a must, especially in the case of the total laboratory automation. In addition to the case of autoreflex and other features, this should be applied for the analysis of the quality control or QC program. In all phases, in a pre-analytic phase, analytic and post-analytic phase, and not limited only to the analytic phase.